record. So question B, the actual diameter of the HCMV is shown in the figure. So the diameter given is actually 0.17 micrometer. This is a very straightforward question. So they ask you to calculate the actual diameter, right, in nanometers. So very easy. They actually ask you to convert only, isn't it? From micrometer, convert to nanometer. Very easy. So to convert, what you do? So this what? Times 1000, right? Or times 10 power 3. So therefore, 0.17 times 1000 is equals to 1, 2, 3. So 170. So 170 nanometer. Okay, or else you should use a computer also can. Computer, calculator. No computer in exam. Okay, C. C suggests the role of viral DNA polymerase within the whole cells. Viral DNA polymerase means this DNA polymerase belongs to the virus. So very easy lah. So the virus DNA polymerase. So DNA polymerase function is to do what? For the replication of DNA, right? So replication of DNA is 1 mark. Is this question 1 mark? Is this question 1 mark? Yes. Very easy. DNA polymerase function is for the replication of DNA, but in more detail, you must know that more detail, let's say 2 marks, right? Okay, you want to know everything. So you know that when DNA replicates, the original DNA double strand will actually you call what? Unwind and unzip. Unzip, the hydrogen bonds broken. So it become 2 templates. Each polynucleotide strand as a template. Polynucleotide strand. Each polynucleotide strand as a template. Okay. And you want to know that how DNA polymerase work. Okay. DNA polymerase, you know that it cannot add a nucleotide to a nucleotide. That means it cannot add 2 free nucleotides. It can only add, so this one is something that you want to remember. DNA polymerase only add a free DNA nucleotide, which you call it DNTP, okay, to a polynucleotide strand. So it can add a free DNA nucleotide to a polynucleotide strand regardless whether the strand is short or long. So therefore, this is something detailed that I want you to know. When the DNA template is formed already, right, because the hydrogen bonds between the 2 strands broke, first thing is that there's another enzyme that will add a very short, give me blue color. There's an enzyme that will add a very short strand of polynucleotide strand on both the template. Okay. So of course, this one must be 5 prime, right? This must be 5 prime. Okay, because DNA will grow from 5 prime to 3 prime. So the new strand must start from 5 prime. So this is called the RNA primer. So both of these are called the RNA primer. Okay, so it acts as a short DNA strand, but actually it's an RNA primer. So then only our DNA polymerase, right, which is actually this enzyme here, DNA polymerase, this one here, then only this enzyme can come in and add the free DNA nucleotide 1 by 1. So give me so now I add the free DNA nucleotide 1 by 1, right? Okay, so like this, okay, this is 1 free DNA nucleotide, this is another one here, this is another one here by complementary base pairing. So this is how the RNA polymerase can do their job. So what does it do? It will add the free DNA nucleotide to an existing polynucleotide strand. So how does it add? By complementary base pairing. And after that, it will have to cause the free DNA nucleotide, the terminal DNA nucleotide here. So this is the last nucleotide here, right? There's one last nucleotide here. This one. Okay, so this is the last DNA nucleotide here. The last one here. So it's called terminal DNA nucleotide residue. it will form phosphodiester bond with this free DNA nucleotide. So the DNA polymerase is to add and to cause phosphodiester bond to be formed between the free DNA nucleotide and the terminal DNA nucleotide of this RNA primer. Okay, and continue to add and continue to form the bond. So forming phosphodiester bond. Okay, so that is DNA polymerase. There's another enzyme that will form phosphodiester bond. Okay, can you tell me what is the another enzyme that will form a phosphodiester bond? Ira? Another important DNA enzyme. Okay, Ira, can you give me the name of another enzyme? Yeah.
Other than DNA polymerase?
Yes. It's called DNA something.
DNA.
Who knows? Anyone of you know? DNA ligase, DNA ligase, okay? Yes, Samantha, correct, DNA ligase. So the ligase function is different. Ligase is to join two short, okay? Join two DNA fragments, fragments by forming phosphodiester bond, okay? So DNA ligase does not join nucleotide to polynucleotide. It joins two polynucleotide fragments together, okay? So there's a slight difference between the DNA polymerase function and DNA ligase function that you need to know very well here, okay? So having explained all this, let's go to the answer. So you see the answer here, state, so they accept the function of DNA, which is to form phosphodiester bonds, because it causes the free nucleotide to join to the existing polynucleotide strand, okay? Reject joins complementary nucleotides. So the main function of the enzyme is not to join the complementary nucleotides, because complementary nucleotides actually can join by themselves. Complementary nucleotides can join by themselves, okay? So this DNA polymerase is to allow after nucleotide form complementary base pair, they want the nucleotide to form phosphodiester bond. So ignore join sugar phosphate backbone. So who joins sugar phosphate backbone is the DNA ligase, because the free nucleotide is not a backbone yet, whereas the two fragments have got sugar phosphate backbone. Okay, so this is where a possible MCQ question can ask you. Backbone is when you got a few nucleotides joined together, it becomes the backbone. Okay, so this is your DNA template, and then this is the new strand, right? So you got two new strand here, right? Fragments. So you want to join them together. So you got two fragments here. Two fragments here, therefore you got two sugar phosphate backbone. So here you got two sugar phosphate backbone. You don't join them with DNA polymerase. You join them with DNA ligase. So that's why here ignore join sugar phosphate backbone. Okay? Because a nucleotide cannot form sugar phosphate backbone. It must be a few, right? So this is first question. So today I'm not focusing on quantity, but I'm focusing on quality. So we see how far we can go for 1 hour. Okay, quality is more important. Your clear understanding is more important than quantity. Okay, now D, the virus in figure 2.1. Okay, let me do it. Okay, the virus in figure 2.1 is drawn as spherical shape. Structure T is always the same shape. What is structure T? Let's see structure T. Structure T. So that means the capsid is always the same shape because capsid is made by protein, right? Okay. However, the electron micrograph show that the HCMV virions are not all the same shape. So what it means here is that the capsid inside the virus that means the capsid, that means the protein coat, is always the same shape, but the virus itself, don't forget the virus that's given there has got a envelope, a membrane envelope, right? Envelope made up of phospholipid. Okay. So it says that, so capsid is the same shape, but how come the virus shape may be different? So they are not all the same shape for the virions, means the virus, because just now in the passage, they explained to us the meaning of virions. Virions are the newly formed virus that are released from the host cell. Okay, so usually they will not just give you a word that you have not learned before and leave it there. They will explain to you if they want to use the word that you have not learned in your syllabus. So therefore, it's actually easy to answer the question one. They will guide you one. It's very fair one, okay? So suggest how the HCMV virions can be of different shapes. You see, today, a lot of questions they ask you, right? You actually know, but the way they ask you is they give you story to bring you to what you learn in your syllabus. What you learn in your syllabus that the envelope, okay, is a phospholipid bilayer. You see, even the mark scheme also say the envelope is what? Is made up of phospholipid bilayer, or it is made up of membrane. You can say membrane, okay? Or you can say that the virus is surrounded by envelope. Okay, envelope or membrane of phospholipid. So you learn that phospholipid bilayer is actually a liquid, like the soap bubble. You blow and you form the bubble. So the coat of the bubble is made up of liquid one. It can move like water one. So the phospholipid molecules can move like water. So therefore, it is fluid. So the phospholipid bilayer is fluid, it is flexible, or the molecules are able to move. That was second marking point. So first marking point, second marking point. So therefore what? So therefore, because it can move, it can easily change shape. Okay, it's like, for example, you try to press this part here, so the shape will actually change, right? Whichever part that you press, it may actually change shape based on how you press it. Probably it will become, you press it here, and it will become this shape. Therefore, the virus may have different shape. Or another thing is, remember every time you get micrograph, right? A scanning electron micrograph or transmission electron micrograph or a light LM, light micrograph, the structure that's been prepared in the slide might have distortion. Okay, for example, the trachea slide that you viewed last week, you see that there are a lot of empty spaces between different layers, like you got the epidermis layer, remember? Epidermis? How can I end epidermis? Epithelium. Okay, sorry. Epithelium layer, and then followed by the C-shaped cartilage ring. Remember, our trachea got C-shaped cartilage ring, right? So, based on theory, these are the layers that we're supposed to see in the trachea. But the slide that you see has got more than these layers, because in between one tissue and another tissue, there's an empty space there. And then in between one tissue and another tissue, there's another empty space there. So why is there empty space there? It's because when we prepare the slide, they might have pulled the tissue and tear the tissue apart. So the connection between the tissue tear apart and leave the space there. Okay, so this is called the distortion. So this is a word, learn up this word called distortion. Distortion during the preparation of the slide. Okay, or the specimen could be squashed when we prepare it. Okay, or artifact. Artifact means maybe some other things not belongs to our tissue, but something else, some stain actually solidify, okay, and form certain kind of structure there. So these are all the things that we can find in our slide because of the defect when we prepare the slide. Okay, when we cut the tissue, okay, we separate the tissue layers. Okay, when we cut the trachea, we separate the tissue layers. Okay, or when we stain the structure, the stain solidify and create certain kind of structure we never learned before. Okay, so this is defect in the slide. So therefore the shape could be different because of that. Maybe for example, or you see something different here, dark color, and then that is actually an artifact from certain stain, for example. Or it could be being squashed when we prepare or being distorted when we prepare. Okay. Number 4 is any other valid point suggest of variation in the tegument protein number or arrangement or organization. So this is what about tegument protein is. You see this particular virus, right? They give you a structure that you never learned in your syllabus. It's called tegument protein here, right? This one here. And the location is between the capsid and the membrane. So it could be the tegument protein that will affect the entire structure of the virus, because let's say if got one side got more tegument protein, it will cause the virus shape to actually become bigger over here because you got more tegument protein here. Use red color. So you got more tegument protein here. So therefore the shape here could be like something bigger here because there are more tegument protein. So that's the answer given there. So all possible logically correct answer will be accepted in this kind of question with the command word of the question suggest. But it's only 1 mark, so very easy to score. Okay. Any possibly logically correct answer will get 1 mark. Okay. Okay, that one is, sorry, it's 2 marks. 2 marks. Okay, E. So let's look at E. With reference to figure 2.1a, state one similarity and one difference between the genetic material of HCMV and the genetic material of typical bacterial cell. So state one similarity and one differences between the genetic material of HCMV and bacterial cell. So both of them, right, either virus and bacteria. In this particular example, the virus uses DNA, so the bacteria also is DNA. So both of them, genetic material is made of DNA. So therefore, the first answer is both have DNA as genetic material. So this similarity, 1 mark only. Then 2 differences, right? One difference only. So any of the difference here will be correct. So we know that bacterial DNA is circular. So this is what you learn in your syllabus. Bacterial DNA is circular. Okay. You see, ignore reference to naked because I think the viral DNA is also naked. Naked means what? It does not wrap around histone protein. Only eukaryotic DNA wraps around histone protein. So you learn and compare between prokaryotic and eukaryotic DNA, right? Prokaryotic is naked. Now, viral one, you never learn got histone, so it's also naked. So they ignore if you say, oh, naked, because we're not comparing between you and pro, we're comparing between pro and virus, prokaryote and virus. Okay. Number 3, bacteria DNA may present in the form of plasmid, whereas virus don't have plasmid. Bacterial DNA has genes. Now, has genes, I don't accept because viral DNA also got gene, the gene coding for virus, viral protein, viral enzyme. So if you say has gene, I will not give mark. That's why you see here, they put here comma. Gene for what? Gene for metabolism. That means the gene inside the bacterial DNA allows the bacterium to carry out metabolism. So it codes for enzymes that can catalyze reaction in the bacteria, whereas the viral gene can code for enzyme but cannot carry out metabolism in the virus. It code for enzyme, or code for protein that can only be produced in the whole cell. So in this way, your answer must be very crystal clear. So you have to read the mark scheme correctly, right? Bacterial DNA got genes, but viral DNA also got genes. So why is this mark scheme a correct answer? Is because we are talking about metabolism, gene for metabolism in bacteria, but gene don't have metabolism in virus, but it will only be active in host cell. Okay. Number 5, bacterial DNA free in cytoplasm. Yes, bacterial DNA is found in the cytoplasm, but the viral DNA is not found in cytoplasm because virus have no cytoplasm, but it is surrounded by capsid or protein coat or T. T is what? T is what? T is the capsid. Okay, T is the capsid. Okay. Okay, so a lot of things to learn as you do the partial questions here. I really like those questions. All right, so let's move on. If you have any question, please stop me and ask, okay? Quality is more important than quantity today because it's our weekend, it's precious weekend. So we want to ensure that the 1 hour span will help you to score extra 5 marks in exam, extra 5 marks. Okay, so from B become A, from A become A star. Okay, so time well spent together. Okay, so F, HCMV is known to infect some types of human cell that carry out mitotic cell cycle. Well, I really like the way they designed the question, man. They link from one topic to another topic. Okay, studies have shown that the presence of one tegument protein, so remember just now that tegument protein, okay, the protein between the envelope and the capsid, right? Okay, so they name this protein UL69. Okay, don't care about the name, they just like to name it. So the cell cycle stops in G1 phase. So that means to say that when the virus infect the cell, the tegument protein of the virus, which the scientists name it as UL69, will actually stop, okay, the mitotic cell cycle of the whole cells in the G1 stage. That means when the cell is in G1 stage, when the virus infect the cell will stop at G1 stage. So the cell cycle will stop there. Okay, so the question asks the student to outline the effects of the presence of UL69 will have on the normal activity of the mitotic cell cycle. So actually the question is very easy. The question is only asking you, how will the mitotic cell cycle be affected when there's a chemical that actually inhibit the mitotic cell cycle at the G1 stage? That's all, very easy. So how does it affect the cell cycle when the G1 stage has been stopped by a chemical? So very easy, right? So that means you need to know based on the cell cycle. So do I have a cell cycle here? I want to take out the cell cycle so it's easy for you to know. Okay, can documents, notes, AS, mitotic cell cycle, right? So we want to look at what happened in a cell cycle, okay? And here, okay? So you learn from here the cell cycle, right? So a cell cycle starts after a cell is newly divided until it divides again to form 2 daughter cells. So it involves G1, give me So it involves interphase. So 80% of the cell cycle is interphase, followed by 15% is mitosis and 5% is cytokinesis. So you need to know that cytokinesis, it is not mitosis. Cytokinesis is after mitosis because mitosis is nuclear division. So during interphase is further divided into G1 as G2 phase. So you know that after a cell is formed, it's very small. Okay. And then during interphase is where a lot of synthesis, a lot of metabolism happen. so that the cell synthesize a lot of enzymes, synthesize a lot of organelles, a lot of structures, and this is where the cell will grow in size, okay, by taking in water and by synthesizing a lot of chemicals that the cell need, including enzymes, including membrane, including organelles, DNA, and so on. Okay, so during G1 phase, it's a growth phase. Growth phase means the cell will grow bigger because the cell synthesize RNA, synthesize protein. That means to say there's the transcription and translation happen to synthesize protein, and this protein can be enzyme for metabolism. This protein can be structural protein. For example, microtubules is structural protein, right? If you talk about muscle cell, actin, myosins are structural. It could be an antibody as well. Okay, it depends. Okay. So growth phase, G1, followed by S phase is where DNA replication occur. Histone protein is also synthesized here. So then G2 phase is the growth phase continue. So the RNA protein synthesis continue. Then this is where some organelles will replicate. The centrioles replicate here. The organelles will replicate here like mitochondria, chloroplast, to prepare the cell for the second mitotic division if there is. But when some cells mature and take up different structure and function, they will no longer go through a second mitosis already. That means fully grown. Okay, so cell size increase during interphase by transcription, translation, and then synthesis of protein, synthesis of phospholipids, synthesis of organelles, okay, in the cell. So let's go back to the question, okay? Go back to the question. Right. So outline, give me the, okay, outlined the effect, the presence of UL69 will have on the normal activity mitosis cells. Okay, let's look at, so how, when G1 is affected, how does it affect the cell? So let's look at the answer here. Ignore transcription and translation. So they want you to write your answer is as in refer to the mitotic cell cycle. So your answer must mention about which phase of the cell cycle, what is affected in that particular phase. Okay, so let's look at answer number 1. So there will be no, what are the phases that will stop? So G1 phase being affected. So if you write G1, no more mark because they already said G1 phase affected. Okay, so you write some other phases that's not in the question 1. Because when you write something in the question, you will not get marked because it's given. Write something else that will also apply, but not given in the question. S phase will stop. G2 phase will stop. Mitosis will stop. Cytokinesis will stop. This one is 1 mark. So this is cell cycle, right? So during G2 phase, what happened? G2 phase, sorry, S phase stop means what? DNA replication will stop. Okay, histone protein synthesis will stop. Then number 3, this one is what I want you to learn. Failure to pass a checkpoint. Now, what is checkpoint? During DNA, let me use blue color, okay, blue color. So during DNA replication, okay, after DNA polymerase, add the free nucleotides. So you form the new polynucleotide strand, polynucleotide strand. So this is called the complementary polynucleotide strand. After that, right, there's a checking of mistakes. So there's another enzyme that will come and move along the DNA, move along this new polynucleotide strand to check whether there's a mistake. That means they add in a wrong base. So instead of adding A, supposed to add A, but DNA polymerase at the wrong, let's say a G, then it will check, oh, there's a mistake here, then they will remove the G and replace it with an A. So this is called checking of mistakes. This is something that you didn't learn in your A-level syllabus. But some students who know it, then they'll say, oh, because there's no S phase, so therefore we cannot check any mistakes or any error that occur during DNA replication. So no error checking occurs. Okay, so error checking during DNA replication. Okay, so this is something extra lah. So don't worry if you do not know, because this is something extra that a student who read other books from internet and write this, they may get the mark. But this is correct. This is correct. But it's not necessary that you know, because it's not in your syllabus. Okay. Number 4, cell. So because of that, cell does not grow, because interphase is where the cell grow in what? Grow in size, right? What do you mean grow in size? So a cell that's newly replicated, right? For plant cell, it is actually square. But when the cell elongate, you can see that the cell will become longer and longer. So you see the width of the cell is the same, right? But it becomes longer. So this is when the cell goes through interphase. This is for plant cell. Animal cell could be different because animal cell can be spherical. So from spherical become bigger spherical and bigger spherical. Okay. Unless you're talking about cuboidal cell. Cuboidal cell become columnar. Must be very careful if they give you a scenario. Okay. Number 5. Idea that the cell does not duplicate or organelles extra material. So same thing, when it doesn't go through G2 phase. So G2 phase is where some organelles will be duplicated, right? So organelles are not duplicated or synthesized. So we can also use the word synthesize. For example, the accept centrioles do not replicate, microtubules that are not synthesized. I think this one is prophase lah. Prophase is when microtubules are synthesized to form spindle fiber, right? So that is actually correct because no more mitosis. Reject if stated as occurring in S phase, even if example G2 given as well. What does it mean? So that means the student say that organelles do not replicate in S phase is wrong because S phase is DNA replication only. And what else? Histone protein is synthesized. That is when something that you write is partially correct, then you will not get the mark. It must be 100% correct to get the mark. Okay. So that's the question. Now let's go to G. G, after a person has been infected with the HCMV, the virus remains dormant state in the body for life. If the virus becomes active again, that means being reactivates, the virus will only cause serious illness if the person has a weak immune system at that time. That means after the virus infect the person, it will remain dormant and the person will not show any symptoms. And even if the virus is reactivated, it will only cause serious illness if the person's immune system is weak. So that's a keyword here. Weak immune system is the keyword. Okay. So now suddenly from virus, they ask you membrane. From membrane, they ask you about mitosis. From mitosis, they jump to another chapter. This is about immune response. This is a very good question. Now, one question asks you a few chapters. Okay, so don't have to worry because you know that they're asking you immune system chapter. They can only ask you what you have learned. Very easy one. Okay, so you must have that kind of confidence. Once you have done your homework, you have studied everything, you must have that kind of confidence one. Okay, so explain why the response to reactivated HCMV is more likely to cause serious illness in a person who has weak immune response. Very easy, right? So when a person's immune response is weaker, why the person can be ill? That's the question. Immune response is weak, person can be ill. So your answer must start with what do you mean by weakened immune response? So you must tell me about what is it that doesn't work that in normal person's body with strong immune response work, and how this will lead to the person becoming ill or sick. So why a person is becoming ill or sick? You learn that it's because the number one, the pathogen can multiply in the body. And number two, the pathogen can actually, number one should be spread, right? Spread. So spread in the body. Number two is it can multiply in the body and not being killed. For 1 and 2 to happen, basically is because not being killed by the immune system. Therefore, they can start to move around in our body through our circulatory system. So they go to the blood and transported by the blood and go to other parts of the body, right? So from the lung, they can enter your ciliated epithelial cells and then go to the bloodstream, and from bloodstream go to the brain, go to the kidney, go to liver. Why? Because our immune system cannot kill them. So they survive, they spread in our bloodstream. And then when they survive, they also multiply, because all living things you learn in biology, when they survive, their main purpose in life is got two. Number one, survive. Number two, reproduce. That's all. So this is the most basic duty and responsibility of a living organism based on biology, survive and reproduce. So immune system cannot kill them. That's what they do. They survive. After that, what they do, they reproduce. After that, what happened? They reproduce. Their number increase. Number increase. Number of the pathogen increase. Okay, so basic knowledge. So now come back to the question. So give your answer. Very easy. Weaken. So you must start with weaken immune response. So you explain to me what do you mean by weaken immune response, and followed by how come the person can be sick. So weaken immune response got a few things, right? You use what you have learned in your syllabus. It's actually very easy to score the full marks one. So let's go to your syllabus. When you learn about immune response, is this one here, right? This one. Remember? What is immune response, right? Remember? So always go back to your syllabus, because they cannot ask you anything other than your syllabus one. So go back to immune response. So what have you learned about immune response? This one is what happened. So your immune response require, number one, your phagocyte to go and phagocytose the pathogen, right? This is step number one, correct? So after phagocytose the pathogen, you know that there are two possibilities, because you have learned two types of phagocyte. What is number one? Phagocyte, neutrophil, right? Neutrophil, and the second one is macrophage, right? Macrophage, the big eater. So you learn two types of phagocyte. Neutrophil, after phagocytosis, they will die one. They die and they form pus, right? You learn about this. Whereas macrophage, after they phagocytose, they are big eaters, so they're stronger. They will move around the body and present the antigen, right? So this is called antigen presentation. So present to who? So now you learn that, okay, if you say weakened immune system, probably your phagocyte is very slow to detect or is unable to bind to the, the phagocyte is unable to phagocytose the pathogen. So that's why it's weakened, right? So it could be phagocyte cannot phagocytose. So any steps that's involved here that doesn't work, then we can say that our immune system is weakened, right? What are the steps you have learned? First step, phagocytose. So cannot phagocytose. Who cannot phagocytose? Neutrophil, macrophage. Then if they can phagocytose, maybe the second step failed. So what is the second step? They are unable to present the antigen of the pathogen, right? Antigen presentation. So very easy. So say cannot present antigen, therefore immune system weakened is because of this, right? Cannot present. Cannot present to who? Present to, okay, T helper cell and B cell, right? So when cannot present, then the next step cannot happen. What's the next step? Clonal selection cannot happen because they cannot present the antigen. No antigen for T helper and the B cells to bind. So no clonal selection. So when there's no clonal selection, next step cannot happen. What is it? No clonal expansion. What do you mean by clonal expansion? So the T helper cell cannot divide and differentiate. So no memory cells formed, no active T helper cell formed, no lymphokine or cytokine secreted, or the B cells not selected. So no clonal expansion means B cells do not divide to form memory cells and plasma cells. So you see, they're actually asking you all these steps here indirectly. What do you mean by weakened immune response? So you have learned immune response is this one. Every single step here might fail. So you must tell them what are the steps that possibly fail. What cells, what step, you will get the mark. Okay, so this is first part of the answer. So when all these steps here fail, is it like antibody not produced against that antigen, right? So this is first part. Then how does the failure of this immune response lead to the person being ill is the second part of the answer. So if you have talked about, oh, this immune system is weakened because of all these steps, you cannot get full marks yet because the question asks you to tell them why the person can be ill because of weakened immune response. So you still have to describe the part where the person becomes ill, why the person becomes ill. Okay, so that's why it's very easy to score the mark if you answer the question. They will guide you to answer unless you focus on one part, half the question, and you ignore the other half. So how to talk about the person is seriously ill is when the pathogen cannot be killed, right? So not phagocytosed, right? Phagocytosed, no antibody bind to it, right? So therefore, not removed by phagocyte, right? So therefore, when not removed, not killed. So not killed, then what happened? Not killed means what? They will start to multiply, or we say, I don't like to use the multiply, the cell will divide. The cell, or if you say virus, cannot say the cell, right? So the pathogen will divide in the body, increase in number, okay, and then spread throughout the body. And they will actually, let's say virus, what do? Virus will infect the cell, so they will damage the host cells, right? Or they can secrete toxin, right? They can secrete toxin and they can affect metabolism of our host cells, right? They secrete toxin may act as inhibitor of some of our enzymes. Okay, so these are the second part to explain the meaning of why the person becomes seriously ill. So when you explain these two parts, you get the full marks. So you really need to pay attention to the keyword. What is it the question want me to answer? Starting from weak immune response is what I need to talk about. What do you mean by weak? And then followed by serious illness. What do you mean by serious illness? Okay, so let's look at the mark scheme. So mark scheme number 1. So there's a time delay in immune response. That means after the pathogen is activated, our immune system is very slow in the response. So in response, like what? Slow in phagocytosis, slow in clonal selection, clonal expansion. So say take a longer time for all this process to happen. So slower immune response. Okay, slower immune response. Slower immune response, or only primary immune response can be activated, no secondary immune response, maybe no memory cells, right? The immune system weakened in the sense that no memory cells, even though this person's infected before. All possibility are correct. Answer number 1 is talking about what do you mean by immune response is weakened. Okay, slower. Okay, then so more time, so slower, so more time for so this is how it causes disease. Slower means what? That means more time for the virus to infect the cell. So when they infect the cell, what do? They replicate in the cell, or they can start to spread. and it can cause damage to other parts of the body, to other cells in the body. Okay. Reject if a student say, or more time for the virus to carry out mitosis, because virus cannot carry out mitosis. So your statement got half correct, half wrong. The whole thing is wrong. So you need to know virus cannot carry out mitosis. One virus got no nucleus, virus not a cell. So therefore, you must write the correct term lah. Okay. Number 3, maybe there are some fewer cells of the immune system in circulation. So why the immune system is weakened? Because it's lack of, let's say, lack of fewer macrophages, fewer neutrophil, fewer memory cells, fewer B cells, T cells, okay, fewer T helper cell, T killer cells, plasma cells, all the cells that you named correctly. No phagocytes also correct. Okay, it can be no or fewer also is correct one. Because weaken, to what extent weaken, we do not know. So as long as it fulfills the meaning of the word weaken, either you don't have or got fewer than normal people. Okay, red blood cell count, no, white blood cell count is lower. Which white blood cell? So if you write white blood cell, they will not give mark. Can you see here or not? Seriously, you write white blood cell means you're giving me an O level or SPM level answer, does not qualify to give you a mark at A level standard. So you cannot give white blood cell when you've learned more than this detail in A levels. You have to give to that detail you have learned in A level to score the marks. This is when the student say, I thought I gave the answer, but I didn't get the mark because you did not give A level answer. You give SPM answer O level, so cannot. You must give A level answer, the one that you learned in A level syllabus, the new one that you learned, not the old one that you learned in last year in your secondary school. So you have to spend time to learn up your A level syllabus content. Very important. Okay. Number 4 and 5, consequence of fewer immune system cell. So just now we talked about there are fewer cells, right? So fewer cells, so what? So you depends on what is the cell that you mentioned, right? If fewer macrophage and neutrophil, give me fewer macrophage and neutrophil, that means fewer chance of encountering with the or phagocytosing the HMC virus pathogen, okay, fewer chance of encountering or phagocytosing the pathogen. If you talk about less memory cells or less B cells and T cells, so less cells that can encounter the foreign antigen, right? Correct? Okay. Less plasma cells. Plasma cells means they will secrete antibody, right? So there'll be less clonal expansion. Why less clonal expansion? Because less antigen presentation, so less clonal expansion. Okay. So all possible answers that are correct are correct. So there are 5 marking points. First marking point is immune system is weakened, so therefore there be delay in response. What kind of response? Why is delay response? Because of fewer cell or no cell. Number 3 is no cell or fewer cell. Number 4 and 5, what is it that is slower? And then when it's slower, number 2 is how come it can cause disease? Okay, the pathogen can divide, the pathogen can spread. So this is a very good question to ask a thorough understanding of this infectious disease and immune response. Very good question. I really like it. Okay, so